Welcome participant. Good morning all. And I think Dr. Jadhav sir also joined. I think he is also in training. So welcome him
Yeah, he's
also.
also one of the trainee. Yeah, Dr.
Yes,
Yadav.
sir. So Jadhav sir, if somewhere I am wrong, please correct me. So thank you, sir. Thank you, coordinator sir, for giving me opportunity. So we will start oyster mushroom production. Though I am seeing lot of faces who are already growing the mushroom. So
Dr.
uh,
Anupam,
yes.
sorry to interrupt. You are having 1.5 hours.
Okay, sir. Thank you, sir. Thank you. It will be
You can,
sufficient, sir.
you can deliver your talk very effectively and with all the explanations so that there is no any confusion with the participants.
So the
And
sir question R is included in that, sir.
eh?
Question answer is
Yeah,
included in
question
that.
only. Yeah, because in the last 15 minutes, you can give 15 minutes so that they can interact with
Okay.
you.
Okay, sir. Okay, sir. Okay.
So
Okay, sir.
thank you very
Thank you,
much.
sir.
Thank you.
Thank you, sir. Thank you. So oyster mushroom production, as you all know, that oyster mushroom is one of the important mushroom. And as Dr. Paul Stamets, who is a
अनुपम
renowned mushroom.
थोड़ी सी आवाज कम आ रही है थोड़ा सा और
Okay,
करिए
sir.
या प्लीज
As you all know, that oyster mushroom is one of the important mushroom. And uh, um, the renowned mycologist Dr. Paul Stamets said that it is only mushroom which is most effective nutritionally and also means uh, bioactively important mushroom. So yes, this is one of the mushroom which balance your production on the way and also balance your nutritive demand, because as you see that all other mushroom having difficulty in growing. If you see in button mushroom, that cultivation is bit not very easy for a initial grower. Similarly, in shiitake mushroom you require a technical information regarding sterilization and other means laminar airflow. So that's is some that means that makes the other mushroom bit difficult. But oyster mushroom is easy to grow, one thing, and it is also a diverse mushroom. If we say oyster mushroom, it is always a group of mushroom. Here there are a lot of species which can be cultivated, and important thing is it is having a certain quality which make it distinct from other mushroom. One is lovastatin. So lovastatin is a bioactive compound which actually a statin group of chemicals which blocks the production of the cholesterol in body. So eventually if somebody is having high cholesterol in their blood, so it eventually lowers down. So similarly, antioxidant, which you all know that it helps to increase the cell viability, and cell death is reduced if you are having a lot of antioxidant diet. Third one is plurin. Plurin is a kind of beta glucan which is actually having anti-cancer property. What it does is it it activates the immune system. So it doesn't absorb in the body, but it enhance or activate the immune system activities. And fourth is d-mannitol. It is another compound which actually helps the patient which are having hypertension problem. So d-mannitol reduces the hypertension problem. And if you have some neurological inflammations, so then for that is gamma aminobutyric acid, which is this compound actually reduces inflammation regarding to your central nervous system. So these are the important bioactive compound which can be grown, which can be taken when in oyster mushroom, and these are the major ones. There are others very small bioactive compound also, like small mycoproteins are also there, which are still being discovered daily on the basis day. I can say daily because mushroom is a superfood, and daily a new compound is coming from mushroom in different mushroom. As you all know that mushroom having huge diversity. Similarly, if we say about nutritive value, so protein percent in oyster mushroom is 18 to 25. That is based on the dry weight basis. So if mushroom having 90% of water and remaining 10% is dry weight, in that dry weight, if we see there is 18 to 25% protein, 49 to 57 carbohydrate, 6 to 15 is crude fiber, and 2 to 3 is fat, and remaining 5 to 8% is ash. Ash is mineral compound, which mean the lot of minerals are present in oyster mushroom. So in totality, we can say that it is a good blend of bioactive compound and nutritive compound. So yes, it is. We can say that it can help in building your good body, good immune system. So overall it is good, good choice for the, if you are a mushroom grower, you can take as a good choice. And not only for grower, if you are a good mushroom consumer, so you can add oyster mushroom in your diet. So one more important thing what we will discuss here is that oyster mushroom can be cultivated round the year if you don't have environmentally controlled plant. Suppose you are not having environmentally controlled plant, and you want to grow mushrooms, oyster mushrooms. So this mushroom is more suitable because there are a lot of species which can be cultivated in different temperature and different environment. So first we will discuss Pilotus aranjai, Kabul dhingri. This is also known as king oyster. Because of its taste, it is said as king oyster because it is unique in all oyster mushroom, and it is very tasty mushroom. If you get chance to eat most of the oyster mushroom, then you will find as a bit tastier mushroom. But one of the problem with this mushroom is this is low temperature mushroom. So the temperature requirement is 10 to 15 degrees Celsius. So this is problem for tropical and subtropical region. But for temperate region, you can cultivate this mushroom. When the temperatures are low, you can cultivate it. And the another problem with this mushroom is that it require a autoclaved substrate. Means the substrate should be sterilized in this mushroom. So now we are now we are trying to develop a technology so that we can cultivate in pasteurized substrate also. But yes, most of the varieties of these mushrooms are cultivated on the sterilized substrate. So you require autoclave, you require laminar airflow. But this mushroom can be clubbed with the shiitake mushroom cultivation, or if you're having spawn facility, then it can be clubbed very easily. So that's why the reason in India it is not much been grown is because of the technicality involved. It means you require pasteurize, you require a sterilizer, it means autoclave, and you require laminar airflow machine. So that's why it is not grown much, and so that's why we can say that this mushroom will have future if this high temperature varieties if we have, and if we have the easy cultivation technology, then this mushroom will boom up, and we are trying for that, and hope so we will get a technology that can be cultivated in a little bit high temperature like 24 and 25 degree, and moreover in pasteurized substrate. But still it is number two mushroom in oyster mushroom, if we say in China, it in all over the globe. So if we say the total oyster production, so out of total oyster production, the number one is Pilotus austratus, and second is Pilotus aranjai. So yes, it is more cultivated in China, and more in mechanized farm it is cultivated. So because of its taste, if you get the chance, you will not forget this mushroom. Once if you eat this mushroom, you won't forget. Second is Pilotus austratus. It is another mushroom which is low temperature loving. So the temperature requirement is for pinhead formation is 14 to 18 degrees Celsius, and for food ball development temperature is 18 to 24 degrees Celsius. So this comes in various colors. So you will find black color, gray color, brown color, lot of color you will find, and you will find as a more bunchy type mushroom and more vigorous mushroom. So this austratus having higher yield also, and moreover it comes in bunches, and it is basically cold loving. So whenever temperature are 24 20 18 so 18 to 24, you can cultivate it. Only thing is you have to see that market you have to see whether which color of mushroom is more preferred in the market, then only you can try any of these mushrooms. So you can see a lot of strains regarding this mushroom. So we also have 5 6 colors, so that depends actually. But in European countries they usually prefer this black to gray color, but in India both basically white color is more preferred. For that purpose, there is one variety called Pilotus austratus florida. It is actually, sometimes it is known as a Pilotus florida, but actually it is a variety of Pilotus austratus. So this mushroom is white in color and also comes in bunches, but the fruit body is bit flat. So we will find a flat fruit body in this mushroom. So that's why its keeping quality is good, because water deposition in the flat fruit body is less. Moreover, it is little bit adaptable because the temperature requirement for this mushroom is it can be cultivated from right from 15 degree to 25 degree. But in 15 degree, the growth is a little bit slow. and in 25-degree you can have a growth little bit faster. So because of its adaptable nature, because of its white color, and because of its flat shoot body, it is cultivated more in India. So this is one of the prominent mushroom which is cultivated in northern part and central part also. Another species is Pleurotus citronopletus. There people also know it as Pleurotus citronopletus variety cornucopy. So variety cornucopy is white in color, and variety citronopletus is yellow in color. So this strain is basically species is basically cultivated when the temperatures are 21 to 27 degrees Celsius. Only thing you have to make sure is when you are packing this mushroom, you have to take a bit care because the cap is very fragile. It will break very easily. So very cap having very crumbly nature means it breaks like a biscuit. So how does the biscuit break if you crush it? So simply this will also break. So only thing is whenever you are growing it, you have for packaging or marketing, you have to take special care. the the flavor of this variety is the species is nutty flavor. So you will find a bit different flavor from Pleurotus austratus, Pleurotus florida. So nutty flavor you will find in this species. One more species is there, Pleurotus pulmonalis, also known as Pleurotus sajar kaju. So this is basically a soft texture mushroom. If you compare all other mushrooms, so it is basically soft in texture, and moreover it is a little bit can be grown in the subtropical region. So subtropical region means the temperature requirement is 24 to 28 degree. When I am saying the temperature requirement is 24 to 28 degrees, then it is indoor temperature. So outdoor temperature may be 5 to 10 degree 5 to 7 degree higher in summers, and in winters 5 to 7 degree lower. So you have to focus on the indoor temperature. So the indoor temperature is 24 to 28 where it can be cultivated. Only thing is there are in this species there are two type of varieties are there. One is bunchy type and another is non-bunchy type. So some varieties will come in bunches, some variety will not come in bunches. So for that regard, you have to know which variety is having bunchy type and which variety has having non-bunchy type. If it is non-bunchy type, then you have to remove the polythene sheet. If it is back cultivation technology, then you have to remove the polythene sheet. If it is non-bunchy type, then you can take the fruit body from holes or the slits you have made in the bags. So in both case, you have to remember these things like when to remove the bag and when to make the hole in Pleurotus pulmonaris. Another one is Pleurotus jamor, that is actually pink oyster mushroom. So uh, this mushroom is um, pink in color. So also known as Pleurotus eos. So eos also, and there are two three names for this mushroom. And but the main distinguished character is that it is requiring high temperature. The temperature requirement is 24 to 32 degrees Celsius, and the strain the variety the species is pink in color. The temperature requirement is also high. Moreover, the spawn time is also less. So spawn duration is 7 to 10 days. The spawn will be completed in 10 days, and you require 13 to 14 days for fruiting. So 13 to 14 days you will get this quality of fruit, means you will get this size of fruit. And one more important thing is where temperatures are low or spawn rate is less, then it will take again 25 days, 20 to 25 days. Remaining all other oyster will take around 20 to 22 days, depending on the species. But there are variation in varieties, so some varieties can be little bit earlier also. But uh, for a pink oyster mushroom, this is the general trend that it will take around 13 to 14 days for flush first, and seven after 7 days again the second flush, and after a third flush will be after 7 days of second flush. So it will take around it will take the whole cycle will take around 40-45 days to complete. But in other oyster mushrooms, so the first flush will come around 20 to 25 days, second flush will take around 10 to 12 days, and third flush will take around 10 to 12 days. This is the variation, so it is fast growing. So one another species, Pleurotus membranaceus. So this is another high temperature species, but the fruit body is bit thin here. So as you see the name membrane caseous, so like membrane type of fruit body will be there. So thin fruit body will be there, but the temperature requirement are is higher again, and the spawn duration days are again 7 to 10 days, and it will take around 10 to 13 days to fruit. Only thing is this species in white in color. So the drawback, sometimes some grower ask that they don't want a color variety. For that, this is one of the suitable choice, that if they don't want a color variety and they want a white variety, so they can cultivate it in summers. So beyond 32 degree, we don't have till now we don't have any cultivation technology for a high temperature, means beyond 32 degrees Celsius, but we are finding it. So till now, so Pleurotus up to 32 degree, you can cultivate the oyster mushroom. After 32 degree, you have to cultivate milky mushroom. If you are growing in natural conditions, means no environmental control conditions are there, so then you can cultivate these mushrooms. Um, when we discuss about this species, we should know about the biological efficiency also. So biological efficiency, because we are here, we are dealing with genetic improvements of mushroom also. So we are developing the varieties every year. We are testing the varieties, and we are developing the varieties. So biological efficiency is not static once; it will improve means within some time gap. But a general biological efficiency for a species is fixed. So like for Plotus kaju, it will be 40 to 80%, but few strains are there that can beyond it go beyond 80% also. So but what my point to uh, what my point here is that Plotus arenji in still in India having low biological efficiency when we are not adding any supplement. This all the data is when we are not adding any supplement. So Pleurotus arenji is still having lower yield, and means 20 to 30%. And moreover, this biological efficiency, those who are new to the mushroom, is that it is a like a biological efficiency is like a unit for mushroom. Like in crop plants, there the unit is like kg per hectare or quintal per hectare. So in mushrooms, it is like biological efficiency. In button mushroom, the biological efficiency is calculated on the compost. So there the biological efficiency comes around 20-25% or even sometimes 30%. So this is the biological efficiency in button mushroom, but there we calculate on the wet compost. Here we will calculating on the dry substrate. So that's why the biological efficiency is bit higher. This is not because of the high yield, actually this is because of the calculation what we are doing here. So in oyster mushroom, we will calculate the biological efficiency from the dry weight of the straw. Why we are doing this? Because we are not adding here any supplementation. If we are adding some supplementation, then also that that will come into consideration. But when we are not adding any supplementation, so it is better approach to cultivate from the dry substrate. Why I am saying? Because moisture can vary. Moisture, it can be like sometimes 67%, sometimes 68%, but dry weight will not change. So that dry weight will not change very easily. So yes, we can. Then we have a very clear idea of biological efficiency. So that's why we are calculating here in dry weight. So, what are the four important steps for oyster cultivation? As I already said, that cultivation is very easy once, and it doesn't require much effort. So, you require four important things. One is you require substrate on which the mushroom will grow. Second, you should know the technique from which technique we will make the substrate pasteurize or sterilize. Third requirement is spawn, and fourth is sanitation. For successful growing, these four components are required. So first is preparation and procurement of spawn. As you all know that spawn is the seed of mushroom. So you require that seed. For that seed, you can contact the government agencies. So you can visit our DMR website. There you will find a lot of equip centers are there. So there are around 30 to 32 equip centers are there. So from there you can take that address, and you can ask from them if they have mushrooms, if they have the spawns. And also you can have the spawn from private producers also there. So you can have that spawn from there also. So but the spawn procurement is why it is so stress, is because like in wheat you will find the spawn easily when you go to the shop, but here the spawn is as the durability of spawn storage is not very high. So more than 3 months is not possible to store the spawn. So that's why for the spawn producer it is a big challenge. So you have to ask whether that spawn is present or not. If you are planning like 20 days after you are planning, so you can ask that, then it will be freshly available to you. So for that, the preparation and procurement of spawn is required. Second is substrate preparation. So substrate preparation is, in India it is mostly cultivated in wheat straw and paddy straw, but there are other substrate also where it can be cultivated. So that substrate preparation is which type of substrate you have to take, and what at what size it have to be cut. Third is spawning of substrate, and fourth is crop management. So these are different centers, and these are different private spawn producer which are present in India. The list can be provided by Dr. Gautam. So Dr. Gautam or your coordinator Dr. B. L. Atri sir. So these list you can take and you can ask for the spawn. Second is substrate preparation. So substrate preparation, three things you have to remember when you are choosing any substrate for oyster mushroom is that it should be cellulose, lignin, and hemicellulose rich. So that information you can find in easily in different website, that how much percent of cellulose, lignin, and hemicellulose is present in agriculture waste. So these one point is this. Second point is, if should it should be devoid of any resin compound, resin, gum, this type of compound should not be there. It will affect the spawn. And third is that it should have the nutrition nitrogen content from 0.6 to 1.5%. In between it should have. If more nitrogen is there, then chances of green mold are there. If less nitrogen, 0.6%, if less than 0.6% nitrogen is there, then again the chances of spawn is very much low. So these point is to be remembered when you are choosing any substrate. So for if that's a study was conducted, and it is shown that more than 200 type of different agriculture waste can be used for the cultivation of oyster mushroom. So you can use means leaves of the trees also you can use. You can use sugarcane bagasse also you can use. So different type of agriculture substrate can be used. So when you have selected the substrate, so you have to know the method of pasteurization or sterilization, because you cannot directly use the substrate. If you are using directly the substrate, what will happen, you will get the contamination of competitor molds. So there are chances that you won't get the full biological efficiency, or in some cases you won't get anything also. So there are 2-3 methods which are important. One is hot water method. So that is very easy approach, and can be started if you are making a very small, not very bigger farm. If you are making very small farm and you require a daily 20-40 bags daily of cultivation, then you can use this method. Second is tunnel method, and third one is autoclaving. So first we will discuss hot water treatment. So in hot water treatment, we will take straw and we will cut it. We will chop the straw 5 to 10 centimeter size. We will cut it. And if we are using less than 5 centimeters straw size, then what will happen, the bag will become compact. So more bag will create a problem in pinhead formation. So that for that, what you have to do, you have to means you have to take the average, and you will find that 5 to 10 centimeter straw is available size, then you can use it. Then second step is soaking the straw. You can directly soak the straw in hot water. So in paddy straw mushroom, you can in paddy straw can be soaked for 70 to 80 degree for 45 minutes. And actually somebody is chatting that slides are not visible. Is it that?
Sir, all good, sir, all good.
All good. Okay. So if we are soaking the straw for paddy straw, we will soak for 70 to 80 degree for 5 minutes. Why? Because paddy straw having silica content, high silica content is there, so it will take high temperature to pasteurize it. But the time requirement is less, because after some time of soaking, the paddy straw will take a lot of moisture. So that's why the time is less, but temperature is high. But in wheat straw, you can soak for 65 to 70 degree Celsius for 60 minutes, and excess water is drained, and after that it is put it in perforated surface. In any perforated surface you can put it, and after that you can use that substrate for spawning when the moisture is 65%. So the layman method of testing the moisture percentage, you press the straw and your hand get wet and there is no drops coming from your palm, that is actually 65%. But you have to take care in paddy straw, because paddy straw, if it is wet more, then the spawn will not be that good if you are not maintaining the um, not maintaining the proper moisture in paddy straw. So that 65% moisture will achieved after 3 to 4 hours. If it will depend on your humidity percentage outside. But yeah, for drying the straw to 65%, it may take around 3 to 4 hours. You can also use spin dryers. Also, there are spin dryers. It's spin dryers you can use, or you can use the press dryers. So spin and press dryers you can use to dry the moisture, to take the moisture up to 65%. So here we will take the straw, or we can take any container where we will dip the straw in hot water, and after that, after drying it to 65% moisture, we can spawn it. Second is steam and tunnel pasteurization. So this is another approach of pasteurizing. So it is done when the quantity are large. So suppose you are cultivating around means 500 kg or 600 kg daily. If you are cultivating, then you require this tunnel pasteurization. So tunnel is a enclosed structure, and it is basically the steam supply is there, and there is also a blower facility is there. So steam and blower facility will help that um, making the temperature even in the straw. So what we do here is, first day we will wet the straw. So it will take around 24 to 36 hours to complete wetting of the high quantity straw, like 500 kg, 600 kg if you are using. Then it will take around 24 to 36 hours to wetting, and also it will require regular turning, churning also it will require. So after doing this, we will add 1% calcium carbonate. Calcium carbonate normal grade we will use, and we will use 1% calcium carbonate in that, on the basis of dry weight. So initially, if we have taken 500 kgs, so after wetting it, that it will come around 1500 kgs. So 5 kg of um, calcium carbonate is added, so that we can balance or adjust the pH. Why we are doing this is because Pleurotus prefers a slightly alkaline substrate. So if the substrate is 7.5 or 8.5, it will help that oyster mushroom to grow, because oyster mushroom can cultivate up to 10 pH also. High pH is will not give the any altered effect in oyster mushroom, but what will happen that green mold and other mold will not prefer this high pH. So that's why if pH is 7.5 or up to 8 is, that is sufficient for cultivating oyster mushroom. And also um, it won't allow other mold to means come in your bag. So that is one advantage. After that, we will make the pile, and we will keep it for 1 day. So first day we have wet the pile. First day we will do the wetting. Second day we will make the pile and add calcium carbonate. Third day we will keep the gap. Fourth day we will do the turning. Fifth day we will keep the gap, and sixth day we will put in the tunnel. So here we will do the pasteurization step. So first step is preconditioning, or before pasteurization we will do the preconditioning. So in that case, we will make the temperature up to 48 to 52 by the use of steam for 2 hours. Why we are doing this? To increase the temperature evenly in the straw, because the quantity of straw is higher. So that's why we will make a even temperature 48 to 52 degree for 2 hours. And after that, we will do the pasteurization. So this is the actual pasteurization temperature, 52 to 62 degrees Celsius for 4 hours. You can, if you have a good facility, and if the temperature is reaches quickly, then you can directly go to the pasteurization step also. Directly 58 to 62 hours you can do. But better if you eventually, if you increase slowly, if you increase, then the efficiency is more. So post conditioning is 48 to 52 hours for 2 hours. Then after that, pasteurization 58 to 62 degrees Celsius for 4 hours. After that, uh, we will cool it down gradually. So it will take around 36 hours to slowly we will cool the our straw. Directly also we can cool it, means directly we can cool it, and we can spawn it. But there are studies like if you do the conditioning, if you gradually cool the straw, then you will get a bit higher yield. So that's why it conditioning, what it does is it weakens the straw structure, it weakens the cellulose, and little bit thermal degradation is there. So that will allow the mycelium to easily penetrate the straw. That's why we are doing the conditioning for 36 hours. And last technique is sterile technique. Actually, this technique is basically used for the Pleutis aerangei. As you know that Pleutis aerangei require sterilization. So for sterilization, what we will do, we will soak the straw in the cold water. We will add 20% wheat bran on the basis of dry weight. We will add 20% wheat bran, and we will fill the straw and substrate and the wheat bran in a heat. After mixing, we will put into the heat resistant polypropylene bag. The size can vary around 2 kg, 1.5 to 2 kg size. We can use it, and then we will sterilize in a autoclave around 121 degrees Celsius for 22 at 22 pound pressure for 2 hours. After that, we will take it out, and we will allow it to cool for 24 hours. It won't cool quickly. We have to give 24 hours to cool it. And after that, we will spawn in laminar airflow. Outside spawning is avoided in autoclaving method. For pasteurized method, you can do the spawning outside the laminar airflow, but for sterilized substrate, you have to do the spawning inside the laminar airflow. Third step is spawning. Now, from all the technique, if you get this straw, sterilized straw or pasteurized straw, then we have to do the spawning. So in pasteurized condition, when we are doing the spawning, we require a room which is pre-fumigated. So pre-fumigated room for means if a room which is fumigated 48 hours before by use of 2% formaldehyde is used. So spawning is done in that room only. Open spawning is avoided. In long run, if you are doing the open spawning, then you will find the frequent contamination in bags. So it is better if you are using a enclosed room, and it should be pre-fumigated. The spawn rate in the oyster mushroom is 10% on the basis of dry weight. And if you are using on the basis of wet weight, then it is 2.5%. Then after filling the bag with substrate and spawn, then you can that bag, if you can use the bag, polythene bag, or you can use the trays, you can use the bottle. So all these things can be used in oyster mushroom cultivation. What we are doing is, here we are taking 5 kg bag. You can take any size from 3 kg to 7 kg. You can take any size, but we are using here standard 5 kg bag. The polythene bag what we are using is 18 into 21 inches size, and the micron is more than 51 micron, and the gauge is around 200 to 300 gauge. So this polythene bag we are using. We are also using trays also, and bottle cultivation we are using for Pleutis aerangei only. So uh, these methods can be used for the cultivation of the substrate. So when we are making the polythene bags, if it is a bunchy variety, like in Pleutis florida is there, or Pleutis austratus is there, so then we are making 10 to 15 holes from all the side. That hole should be equidistant. It should not be one hole should be close, another is far. Like that, we are not making. We are making the equidistant hole, and two holes we are making at the bottom of the bag, so that excess water is drained out. So there are two type of spawning, but which is followed in oyster mushroom and milky mushroom also. So there are two type of spawning. So one is mixed spawning, which is also known as thorough spawning, and second is layer spawning. So both the spawning method we are using here. There is no significant difference in yield while we are using the mixed spawning and layer spawning. Only thing is, in layer spawning we can compress the bag more. So what is mix spawning? We will mix the substrate and spawn on the 10% on the dry bed or 2.5% on the wet weight, and we will fill the bag. Second is, in layer spawning we will make the layers of spawn and alternate the layers of alternate layers of substrate, spawn, substrate, like 3 to 5 layers we will make of, and we will fill the bag. In the case in layer spawning, we will put maximum spawn on the circumference of the polythene sheet, means outside, means peripheral side, and not in the interior side. Outside that, means on the periphery of the polythene, we will add the spawn. And that in advantage with this method is, layer spawning is, we can compress the bag more, while in mix spawning we will make a medium compression. So that advantage we can take in the layer spawning. When the spawn run is over, when the spawning is done, we will keep the bag in the incubation room where the spawn run will occur. So here we will maintain the temperature so that the mycelium can incubate, and the mycelium can evenly form around the substrate. So that is actually incubation. And here we will max, in most of the floaters case, the temperature is around 24, but you can increase the temperature in floaters jammer up to 30 degree also. And similarly in Sajar Gaju, up to 26 you can go. So it will depend on species also. So incubation temperature is around 24 to 26 for means summer species, or 28 to up to summer species, and for winter species around 18 to 24 degrees Celsius. So this is the incubation temperature. After that we will induce the fruit body, and these are the different means parameter we have to follow. We will discuss it. First is incubation. In any case, while in incubation, the temperature should not increase more than 30 degrees Celsius. That is thumb rule. Moreover, in initial 2-3 days, you have to take a bit special care, because initial 4 days are very crucial. If spawn run wouldn't start, then then the chances of contamination, other competitive mode, will increase. So initial 3-4 days are very important, so that mycelium easily colonize the substrate. uh, if the temperature goes beyond 30 degree, in that case you can add some water on the roof, on the wall, so that that cooling effect can occur. If any case the temperature goes beyond 30 degree and it goes up to more than 12 to 16 hours, then the spawn run will be halted. So that you have to take care. So initial 3-4 days are very important. In this incubation stage, you doesn't require the adding of any fresh air in the room, or the relative humidity, creation of relative humidity is not required, and no fresh air is required. And spray also, any spray is also not required. If temperature are maintained, then spray is not required. So in incubation, just you have to check the bag daily. Second is full body induction. So now if it will take around 15 to 22 days to cover the bag with mycelium, so that if that is over, then you have to start the step of inducing the fruit body. So for that you have to increase the relative humidity. So increase of relative humidity will help to trigger the fruit body formation in the mushroom, because as you all know that mushroom require little bit change in the environmental condition to appear in vegetative, to change from its mycelium stage to fruit body stage. For that case we require a little bit change. So for that we will increase the humidity. Moreover, humidity is also required for maintaining the moisture around 90% in the fruit body also. So that's why the higher relative humidity is required. So in that case, in oyster we require around 80 to 85 or 90% humidity. So for that we can spray the water, we can use the humidifier, or we can use the foggers also. We should avoid the spray on the fruit body. So when the spray is, if we are doing the manual spraying, so more spraying should be done on the walls, on the roof, on the floor, and less should be on the fruit body. If you are spraying and some droplets falls on the fruit body also, after that you have to start your exhaust fan or the fan, or you can give the fresh air, so that fruit body moisture can be evaporated. Otherwise what will happen, the chances of degradation of fruit body, either by bacteria or by excessive water, can be there. So that's why that is more important. Second is addition of fresh air in the room. So up to spawn there is no requirement of fresh air, but since the oyster mushroom mycelium during spawn run can tolerate up to 20,000 ppm of carbon dioxide, but when we are taking the fruit body, then the carbon dioxide requirement is very less. Then it should be below 600 ppm. For that, we have to introduce the fresh air. If we won't introduce the fresh air, the numerous pinhead will form, and these pinhead will also affect your means the pinhead won't allow the fruit body to grow into full mature size. If the size CO2 concentration after if suppose pinhead you have pinhead is good and pinhead has been formed, you have given fresh air good at this time of pinhead formation, but when the fruit body development is there and you are not giving the fresh air at that time, so at that time you will find that pinhead were okay, but your fruit body stem will be longer. So there are two symptoms of less fresh air. One is at pinhead stage and one is at fruit body stage. At pinhead stage, the numerous pinhead will form. That is the problem when you are not giving the fresh air during pinhead stage. When pinhead development is there and then you are not giving the fresh air, then the stipe, the length of the stem of the mushroom will be bigger. That is again, and so cap will be smaller. So you have to give the fresh air during pinhead stage and during fruit body formation at the both time, and it should be 6 to 8 hours depending on the cross ventilation. So suppose you have good cross ventilation, so then the time requirement can be less. But if there is no cross ventilation and you have to use the exhaust fan and other inlet fans to circulate the air in the room, then you require a then you require 6 to 8 hours. But by my suggestion is you give that 6 to 8 hour in breaks, not regularly. You should not give, it should be given in the breaks. Otherwise what happen, whatever the humidity you have maintained in the room, it will change drastically. So that's why whenever you are using, so you can give like 30 minutes or 1 hour means fresh air, and again you can stop. Like that you can provide the fresh air. Second, another important factor is light. So light is important for the cultivation of oyster mushroom. What will happen that light promote the pinhead formation to mature fruit body. Moreover, if light is more, the formation of pinhead are also higher. So so actually the some of the enzymes are activated by this light in oyster mushroom and other mushrooms also, some other mushrooms also. So you can use the light. It should be around 800 lux for optimum production and 200 lux for the around for minimum requirement is there, but optimum is around 800. Some studies are there that in Pleurotus syringae, if we use blue light, so that will enhance the pinhead formation and the fruit body formation. The blue light studies are more in Pleurotus syringae. In that you can use that blue light. And moreover, when you are using this light, the lux intensity you can measure by either by lux meter, or you can use the visual observation also. If you can use the if you can read the newspaper in the room, that is around 200 lux, and 800 lux is when you entering the room and you can see each and every corner of the room, that is around 800 lux. So that much light for 8 to 12 hours should be required for the oyster mushroom growing. After that, hydrogen iron concentration is basically pH. So pH is one of the important consideration. As I already told that high pH will promote the grow will suppress the growth of the green mold. One more advantage of high pH is whenever the mycelium is colonizing the substrate, then the oxalic acid is released. When the oxalic acid is released, the pH of the bag reduces. So that reduction is there, and in first flush it will be reduced around 5.5 to 6.5. In second flush it will reduce more drastically also. So yes, if we are taking little bit higher pH, then what will happen? That pinhead development pH is around 5.5 to 6.5. So that will remain in that range. So that's why we are using little bit high pH, so that that range will not affect it. So the development of pH will happen around 5.5 to 6.5. So that range will automatically achieved when the pH is around 7.5 or 8. One more important thing is post-harvest practices. So oyster mushroom, whenever it is harvested, the spraying is not recommended before harvested. So if you are harvesting, suppose you are harvesting today 11 o'clock or 10 o'clock, so you should not spray at the morning. So you have to harvest it, then only you have to spray it. Because if you spray the water, so what will happen, if it goes into market, it will easily degrade because the water droplets will be there. So that is not recommended. So young mushroom is around 5 to 8 centimeter in size, depending on species also. So some species like Pilotus ostratus, so the size is 8 to 12 centimeter. So bigger size is there. So that you have to check. But the optimum size what we have seen in most of the oyster mushroom is around 5 to 9 centimeter size is there. If the cap is curling upward after a young stage, so that is actually over mature stage. So before that you can harvest it if the temperature is okay. After when the pinhead will take to develop around 2 to 3 days. So around 2 to 3 days it will take. If pinhead is, suppose today if pinhead arrives, so after 3 days you can harvest it. So you have to see whether the temperature is okay or not. If temperature is okay, like 24-degree temperature is there, then it will harvest in 2 to 3 days. If the temperature is lesser, if it around 17 to 18 degrees Celsius, then we can take around 5 to 6 days to mature. One more important consideration is, suppose you are harvesting a bunchy type variety, and in a bunch you won't find every mushroom mature. It won't happen in a bunch. So if 60% mushroom, 50 to 60% mushroom is mature, if they have attained a full size, then you have to harvest a bunch as a whole. So the post harvest life of a bunch is little bit higher than a single fruit body. So yes, you can pack in a bunch shape. You can also pack in a punnet. So these are the some tactics so that you can enhance the means your quality of your mushroom. This is one of the means physiological problem what it occurs in oyster mushroom. That is not any disease, but because of high humidity. So in that case, what you can do is you can add the fresh air in the room so that humidity of room can be lowered, and you will get a perfectly good fruit body. This is basically high humidity. And what had happened is actually the spores they have stick together and they form the thread. One important problem in oyster mushroom is they produce a lot of spore, abundant spore they will produce. And whenever it is full fruit body stage, means whenever the room is having full bags which is having this fruiting means stage is there, then a lot of spore is produced, and this spore will go to your means lungs by breathing, and that may create allergies to various person. Not all the person, but some person will experience the allergies. So that allergies can be minimized if you are using some protective gear, like you are using some respirator type of mask if you are using, or you are using exhaust fan, so that no spore buildup is there. So you are using exhaust fan, means 8 hours before the harvesting, then there will no spore buildup is there. And moreover, some sporeless varieties are also there. We are also working on that, so that also are available, so you can go for that also. These are the mold symptoms. These mold symptom basically happens when there is not very good pasteurization is there. If the proper pasteurization is not there, then this kind of thing will happen. This is crop web disease, so this will come when the humidity is higher, or other things are also there. But most of the when high humidity in second flush or in third flush, you will observe these. So another one is important is mushroom flies. So mushroom flies is again a problem in the summer time. So you can use from the start of the spawner, you have to use this blue light traps, so that these flies cannot be increased. If this flies will increase, then eventually the larva will increase, and these larva will make the holes, and it will not give a very good quality production in the farm, in the mushroom. Also, this larva will also eat your mycelium, so peanut formation is also reduced. So this was all about the production of the oyster mushroom. So coming to the economics, the oyster mushroom cultivated around, it will take around 35 to 45 rupees to cultivate or 1 kg of oyster mushroom, and market rate will depend around 80 to 120 to 130 rupees. But in some metropolitan cities, that may be higher. But The cost of cultivation will range around 35 to 45 rupees per kg, and the selling price, if we do a better market studies, then it will be around 80 to 100 rupees. So for that cultivation, you require a little bit idea of the mushroom farm. What kind of mushroom farm should be there? So it is more technical than button mushroom farm design. Why I'm saying is because in oyster mushroom there are a lot of approaches of growing the mushroom. So first approach is you can cultivate mushroom daily here also, but in button mushroom that won't happen daily. You can't take the cultivation and harvesting, but in oyster mushroom you can perfectly make that cycle. You can daily make the bags, you can daily harvest the mushroom, because there are different pasteurization method in hot water method, or in other, if you are making any chamber of pasteurization chamber, small pasteurization chamber, so daily you can make 50 or 100 bags. But in button mushroom you require a tunnel, bigger tunnel, so in that you have to make once you have to make the uh, that compost. But here you can make daily. So here if you are doing this type of approach, then the eventually their farm design will change, because at that case you don't require a big preparation yard, you require a steeping tank only. And you, in that case, you didn't require any pasteurization chamber, you require a hot water chamber, or a steam chamber, small steam chamber is required. So in that case that farm design will change. So in a general, but if you are cultivating 20 tons per annum oyster mushroom, so it will eventually cost, if all pakka structure we will take into the account, it will cost around 25 to 30 lakhs. It depends on all other if you are using boiler, or if you are not using boiler, that will depend on that. So there are in India there are two type of structures in oyster mushroom. One is RCC and puff based structure, and another is hut model. So this oyster mushroom very successful in puff structure also, and in hut model. Both it is very successful. So either of one you can go. So a low cost hut model, if we are making the size, if we are making 30 into 20 into 10 feet, so it will if we are having 2 to 3 huts, then we can produce around 8 to 12 tons of oyster mushroom, and the durability of this hut will be 3 to 5 years. The size and the cost for making these huts and all room required for these huts is around 3.5 lakhs, and the machinery is required around 25,000. Around machinery is required for these huts, and the cost which is incurred for this cultivation of the mushroom, this around the working cost, that is around 4.5 lakhs. In that we require ropes, polythene, spawn, straw, formalin, calcium carbonate, etc. What are the important aspect while we are making the room is that room should be rectangular, and it is more preferred if the room is twice the long as width. This I am particularly saying with the reference of this puff panel rooms and RCC rooms. In hut, because the aeration is not much problem, so there you can go for non-rectangular room also. There are chances, because the huts are made from locally available material, and there are a lot of porosity in that. So aeration is more in that structure. But in puff panel, you require a rectangular shape, because airflow is maintained by the air handling unit. So there, the more means airflow is required, because otherwise in circular, in a square room, in corners the air is not as much as in the center. So that's why a rectangular room is required. Moreover, the interior wall should be more resistant. If you are using a RCC made structure using any insulation material, so whenever you are making the room from inside, the inside wall should be more abrasion free, so that it can be more resistant also, and abrasion free. And while this floor should be slopy, so that any water deposition should not be there. And in a room there should be foot dips. When you are entering the room, it should be foot dips. The door what we are suggesting in oyster mushroom is you can use a single door, although there are double doors also and single doors also. But a single door, large door is good for avoiding any fly problems also, and easy filling also, so that your movement is easy, and also fly problem is less in single doors. You can use a inner frame door also, a small inside door, so that easy movement should not be there. So easy movement should be restricted for flies and for other insect and for personals also, so that easy movement is not there, and so that more hygiene can be maintained. The indoors there should be a brush script, so that any crawling insect should be avoided in the room, and air curtain and food tapes are required at the every door of their cropping rooms. In cultivation, oyster mushroom cultivation, we are using racks. Racks we can use, and racks for according to the bag size, we will make the tire size. But the size of big rack is around 1.5 meter to length and 0.75 meter width. And the first tire of rack should be above 6 inches from the ground, and the gap from the walls should be 1.5 feet, and for the movement it should be 1 feet. More gap is required in oyster mushroom compared to cotton mushroom, because the aeration requirement is more, and moreover there the gap is maintained. Each bag we have to maintain a gap of 12 inches, so 12 inches gap from each bag. So because oyster mushroom comes laterally out, so that's why 12 inches gap is required. So these are the humidifiers. There are two types of humidifier. One is impeller type humidifier, that is also called as steel humidifiers. So there's actually there is one motor attached, so that it will liberate the droplets. And one is ultrasonic humidifier. So the size of the this impeller type humidifier is droplet size is bigger, while ultrasonic humidifier has smaller droplet size. So ultrasonic humidifier created humidity will be last longer as compared to impeller type. Moreover, the area covered by this impeller type humidifier is 250 square feet. Both are efficient. Both are efficient. You can use both, but on the basis of superiority, then the ultrasonic one is are more superior, and the humidity lasting in this ultrasonic humidifier is for a longer time. These are the meters which are required. So humidity meter, temperature meter, you always required it. So it should be installed whenever you are cultivating. Lux meter and CO2 meter, if you get eventually exposure while cultivating it, you initially get the idea. But in a mechanized farm, I suggest that these two instruments should be there, so that anyone can detect what are the problem. So lux meter can be installed and CO2 meter can be installed. Uh, in oyster mushroom, there are different type of cultivation system on the based on the bag arrangement. So there are rack method, hanging method, tray method, bottle method, log method, and block system. So rack method is you will place the bags on the rack, then you can cultivate it. Hanging method is low cost method. Only thing is there is difficulty while harvesting and keeping the bags. Another one is tray method. So it is reusable method. Trays can be reused every time. So that also you can use. Bottle system is basically for Pelote syringae, and it is more for the mechanized farms. So when the farms are mechanized, you have a filling system and emptying system, then you can use. Log and block system are where the substrate bag is around 10 to 20 kg when you are using big substrate bag you are using. Then it is called log and block system. If you make in the shape of log, then it is a log system. If you are making in the shape of block, that is a block system. So these are more done in the countries where the temperatures are low. So like in European countries where the temperatures are low, there the bag temperature can be raised by bigger blocks and bigger logs, and moreover, in a control plant it can be done. But where the countries are tropical and subtropical, and that block will what will happen, that the temperature of block will increase if you increase the bag size. So again there you required extra energy to cool it down, so that is not much suggested in our Indian conditions. So these are the cost norms. Actually, these are the cost norms on which we develop the techno-economical feasibility report for oyster mushroom. These are basically taken by the government agency, CBWD, and other agencies, so that a specific cost norms are there for developing that. So it may change area to area also. It is not like that, but we have standardized little bit according to the uh, country's requirement. So that these are the cost norm. On the base of these cost norm, we develop the TFR for the oyster mushrooms. So these are the different plant machinery what we use in oyster mushroom. But one thing you have to specific whenever you are asking for a techno economical fizzy report, two three things are very much important. One is which kind of cultivation method you will use. Either you will go for tunnel pasteurization or you will go for daily pasteurization. That means like using hot water method or another systems. Or second thing, whether you will require a hanging method or means because the cost is affected by these. If you are using a hanging method, so you will the machinery cost will be 5 to 6 rupees lesser. That will all will depend on the requirement. So thank you. This is what I have for my presentation. If any questions are there, you are kindly welcome. I think in chat box there are some questions. I will take it. Can you show the image of cropping room? Definitely. I will. What I will do is I will take a pic and I will share it to Dr. Bialatri sir, and he will take give you that. Sir, how to go for small scale startup for mushroom? See, small scale you can start from oyster mushroom. It is a very good mushroom to start a mushroom. If any mushroom you would want to enter and you are new to this industry, then you can start with oyster mushroom because it will give you idea about mushrooms, and it is in small scale. It is feasible, economically viable. We have to see because for that we have to do calculations. It is seen that about 20 tons is economically feasible. So yes, you can go for small scale. To know how to door looks, then again I will share the photographs of that. Oyster mushroom can be grown in pine needles and Bentley found. Yes, we are trying for that, and we are trying for that. Directly, it is bit difficult. Yes, there are reports that directly it is bit difficult in pine needles, but yes, by doing supplementation and doing this different another approaches, we are trying it, and hopefully next year we will able to. So that's why we are trying. And you can contact me after the 1 year so that we can suggest you more clarity in data because till now it is on cultivation only, so we don't have the clarity on the data. So once the data will come and we will share you the details. What is the control measure of controlling small insect being found in the stem? So there will be a class of I think this one control measures of mushroom insect, so then you can discuss. Please express the turning process because so there is for I think you are asking for this tunnel method. So for that, what we are doing, first day we are wetting it. Second day, but we are adding 1% calcium carbonate and making the pile. Third day, we are keeping as ideal. We are not doing anything so that a temperature, little bit temperature raise you will feel. We will see means the temperature will be raised around 40 to 45 degree of pile. And fourth day we will do again the turning so that mixture is evenly again distributed. Fifth day we are keeping ideal, and sixth day we are filling in the tunnel. So these are the only two turnings we are using, although that is not very thumb rule. But what we are actually doing in this process is one is that whatever the you are adding calcium carbonate, that is evenly distributed. Moreover, a little bit pile temperature will increase. So we are literally reducing little bit inoculum outside also. This can clear. Please note all the alternate source of plastic bag. So now we are using, we are planning to use this biodegradable already. I think in Maharashtra, Jadhav sir is there. I think he can throw more light because in Maharashtra, I have seen some people are going for the biodegradable plastic bags. You can. I also in my lecture I also said that you can use trays also. So yes, you can use this biodegradable bag and trays. Both you can use. Are any backfilling machines are there? Yes, backfilling machines are there, and we have purchased one for shiitake. Basically, backfilling machines are there. So we have purchased around 1.5 to 2 lakhs. That is there, but there is some customization is required. Means customization means because that the filling machine having and that there is one instrument which fill with the rate, means there are some rate of filling like 500 bags per hour. So that rate is actually modified, because what is happening, this bag filling machine basically, and they are used more in spices, for filling the spices in bags. That actually modification they are doing, but that modification basically they are using for dry ones. So for wet ones we have to make a little bit modification. And there are other bigger bag filling machines are also there, but that is around costing around 8 to 9 lakhs. That is bigger one. That is bagging, bag filling and sealing also. They will bag fill, seal, and they will do all the thing. And spawn also they will mix. So that is around 8 to 9 lakhs. Minimum capital to required for starting oyster mushroom farming. So minimum capital you can start with any, means you can start means if you have, you can start it. But for cultivation of proper means for a healthy economically healthy unit, minimum 10 tons 20 tons is required. Cultivation of 20 tons is required, and for that a minimum investment excluding land is around 25 to 30 lakhs. Means I am saying of panel and pakka structure I am saying, so that much is required. But if you are hard based structure, you can start from around 3.5 lakhs or 5 lakhs, around 5 lakhs it will, means for construction is required, and for working capital 5 lakhs is required, so around 7 lakhs is required. uh, Up to what temperature we should cool the straw? So, and means ambient temperature, suppose your in your area the temperature is 25 or 30. So whatever the temperature is outside, you can, for the up to that temperature you have to cool, so that you can do the spawning. How long do the oyster mushroom remain fresh? Although there are a lot of technique to make the oyster mushroom fresh for a longer time, but in general, if we are not applying any measures, so it will only, means depends on the area to area temperature. Also, it will depend, but not more than 2 days. Not more than 2 days, oyster mushroom will remain fresh. Otherwise, what will happen? Shrinkage will happen. So it will also depend on the humidity also. So not more than 2 days. Cost of cultivation, I already told, 1 kg of fresh moisture mushroom will cost, means for cultivation it will be costing around 35 to 45 rupees. Can you suggest any other substrate that you can both Oyster and Pesto? You can add, you can use like if pulses straw also you can use, but in the mixture you can use. I mean, directly you can't use it because high nitrogen is there. And you can use sugarcane bagasse. You can use. You can use mustard straw. You can use. You can use dry poplar leaf. You can use. So these you can use. There are a lot of list is there. Lot bigger list is there. You can use it. Mix substrate variable or profitable? Mix substrates, it depends actually. Mix substrates, if you are using, only thing is you have to find what is the nitrogen percent in that. If my higher nitrogen percentage will be there, then the chances of contamination will be there. If CN ratio is perfect, then I think mixed is a good one. You can go for mixed one. Two, three things what you have to see in mixed substrate. One is all means one thing is availability. Second is nitrogen content in that whenever the final substrate, and third is the air spaces. Air spaces in that substrate. It should not very compact ones. Like if you use sawdust as a substrate, so it will take a longer time for spawn. So that is one. Making holes in my well lead to contamination. So now please, yes, it is. I have seen this plugging of cotton in the holes of the. That is good approach. I appreciated this approach. That will help in a longer run. Yes, that is good approach. You putting cotton is a good approach in the holes. Means there is no any scientific drawback in that until unless cotton is means not contaminated and not used frequently in the same contaminated farm. So I think that is okay. How blue light affecting? So there is one question, blue light. So blue light in basically um, is basically what it does is when pinhead has formed. So there is a requirement of energy to form more pinheads and fruiting body to increase the size of fruiting body. So blue light will trigger that genes. Actually, the genes are some genes are there that trigger the genes so that more formation of energy is there, and ATP, that actually the I will say in more scientific word is that is ATP form is more, and that will help in formation of means formation from pinhead to means fruit body, total fruit body, that will help it. So that's why blue light is affecting that mechanism. Hypsizygous is oyster mushroom. Actually, that hypsizygous is actually the different genus. Actually, hypsizygous, and there are some the people say some blue oyster is a hypsizygous. Actually, it is Pleurotus ostratus only. It is not hypsizygous. Hypsizygous is hypsizygous. There are centrally attached. Actually, the stipe is centrally attached in hypsizygous. The stem is centrally attached. So yes, that is actually Pleurotus ostratus only. Hypsizygous, what in India we are using it is Pleurotus ostratus. For 1 ton of substrate, how much spawn is needed? So that is around 10%. So 10% spawn you calculate for 1 ton. I think it is for dry. So 10% you can use. Email ID I will provide. Yes, you can. There are any supplement to increase the productivity. You can use the wheat bran. You can use 5%. You can use 5% wheat bran. You can use because more you can go up to 20% also. But thing is, whenever you are going for high nitrogen percent, their contamination of green mold will be higher, because then you have to take a lot of care. So 5%, 5% is sufficient. Moreover, but if you add the supplement also, so you will find an even crop also. So that benefits are there for supplementation. But what I will suggest is, in winters you can add the supplement, but in summers you avoid. That my general thumb rule is that in winters we can use, and in summers we should avoid. Can soybean straw can be used? Yes, soybean straw can be used, and I will suggest to add. You can use by hot water treatment. You can use it. Soybean straw you can use it during. Yes, then there is one incubation period. Flies may enter, so you can use light trap, or you can use that. You can plug that holes by using cotton also. What happens when an oyster is sucked? I think it should be shrinked. I think so. Shrink is there. I think humidity problem is there. So humidity problem may create this problem. How moles and moles? There will be another lecture for that, moles and. Sir, about chemical pasteurization. Chemical pasteurization, yes, it is one of the effective technique which was used earlier. But since now what the chemical we are using in that chemical pasteurization method, now they are planning. I think they are in proposal to ban it. So that's why we are not dealing with that more. Now we are seeing another approaches of pasteurization. So that we are working for. Is tunnel design same for button mushroom? That will depend on that quantity, on which quantity means how much quantity you are producing. So that tunnel design will depend. We will share that. We will share you if you whatever you require. You just message me. I will share you that. Can soybean straw used for the cut? Yes, again the same question. Sanitization of room will be dealt by Dr. Anil. I think he will be sharing that in detail. He will be sharing. Any low cost steam pasteurization device, yes, you can make it. That low cost steam, you require a steam to be injected in the room in a small tank. But that you have to means see for security consideration. You have to see it should not having it should not be very it's vessel type. It should be like open type, so you can do it in that also. In our area, Saril Kaju grow well, but due to marketing, we are facing. So whenever you are cultivating any mushrooms, means other than button mushroom, you have to do the market survey. That is thumb rule for all other mushroom other than button, because button having well established market, but other mushroom you have to do the market survey first. Number of water spray in the crop room day, you can do 2 to 3 sprays depending on the humidity percent outside. So for if the humidity is around 60-65%, you can spray 2 times. If humidity is 40-50%, you can spray 3 times. The time duration of spraying should be equal. It should not be like 6 hours, after that 8 hours. It should not like that. It should be equal duration. Can we go for round the area cultivation? Yes, the species what we have suggested, you can go for round the area cultivation. Thickness, I have already discussed that thickness of plastic bed should be 250 to 300 gauge you have to take. Can sawdust can be used? Yes, sawdust can also be used, but only thing is you have to go for this that spawn and time will be a bit higher in sawdust. Polythene bag is banned above 51 micron. It is not banned, I think. So above 51 micron you can take. I think other are all repetitive questions, and other questions what I am seeing is more related to other fungus or improved shelf life that will be dealt upon. So I think yes, for fumigate, yes, all other questions are for that only. So I think for my side it is over. Still you have questions, so I will sir will be sharing also, and if I will be sharing my number also, so you can message me. No issue. Thank you, sir. Thank you for giving time. Thank you, sir.